Hello.
Yeah. Good morning, sir. Yeah, we can hear you, sir. Welcome,
Oh,
sir.
thank you.
Good morning to everyone. Before starting the webinar, I request everyone to remain muted, both audio and video, throughout the entire session. So we have an eminent scientist with us. We request everyone to maintain decorum throughout the session. If you have any questions to ask, kindly write it down on the chat box. It will be read out by the host after the presentation to the speakers. I, Dilshad Begum, Assistant Professor, Department of Biochemistry, Indian Academy Degree College, cordially welcome all the participants to an international webinar on trends in biochemical research organized by Department of Biochemistry, Indian Academy Degree College, Autonomous. Biochemistry is the branch of science that explores the chemical process within and related to living organisms. By using chemical knowledge and techniques, biochemists can understand and solve biological problems. Much of the spectacular progress in biochemistry research, whose primary goal is understanding of the fundamental working of living things. With this in mind, we have two talks from eminent scientists from USA on cardiovascular interventions by Dr. Prakash Dampatar, University of Iowa, USA, and understanding synapse development using Drosophila neuromuscular junction by Dr. Soumitra Chowdhury, National Institutes of Health, USA. Now I take this opportunity to welcome our beloved principal, Dr. E. Jerome Xavier, who is always a backbone for conducting such events. I welcome you, sir. Before
Thank
starting the scientific sessions, I request Principal Sir to deliver the welcome address. So kindly deliver.
Dear resourceful persons, Dr. Prakash and Dr. Soumitra, dear participants, faculty members, research scholars, students from all over Karnataka and India. At the outset, I would like to extend a very warm welcome to each one of you, very especially to our renowned scholar and scientist, Dr. Prakash and Dr. Soumitra, for this fantastic webinar on trends in biochemical research. I was very glad to see the topic because both the resource persons are going to deal with two most important physiological aspect of human uh, human, and in a very special manner. These are all not only physiologically important, but more so for the overall personality of every individual's mind and heart, brain and heart. We know one of the mysteries in the study of human beings is the study on brain, and the other one is the study on heart. Besides the physiological aspect that controls the heart, we know heart is a seat of knowledge, affection, emotion, and love. While we always consider brain as a seat of wisdom, knowledge, understanding, reason, and so on. Although there are thousands and thousands of volumes written on both and research being carried out both on heart as well as on mind or brain, we all know it is not sufficient. There are areas wherein we haven't discovered, particularly when it comes to brain. Almost every month and every year we find new research articles being published on the study of brain. And from studying brain, now they are moved into artificial intelligence. So soon we will find the technology and the physiology might be combined together to give us new information, new knowledge, a new way of thinking, not only our life, but also our relationship to the environment and particularly to the technology. I'm sure the speakers with their excellent record in research and research publications will be able to enlighten us, although it is a limited amount of time, still I'm confident they'll be able to inspire us, enlighten us, and give new perspectives on these two topics, brain and heart. Once again, Dr. Prakash and Dr. Saumitra, on behalf of the management, principal, and the faculty members of Indian Academic Degree College Autonomous Bangalore, I extend a most hearty welcome. And over to you, the resource persons. Thank you very much for accepting our invitation and being with us this morning. Thank you.
Thank you, sir. Thank you so much for your welcome, valuable speech. So let's start with the scientific session now. Okay. We have two sessions. Let's start with the session 1. It's now my great pleasure to introduce Dr. Prakash Dodpatar, our first speaker for this webinar. Dr. Prakash Dodpatar graduated from Karnataka Science College, Dharwad in 2005, pursued post-graduation in biotechnology from Karnataka University, Dharwad in 2007. He received his PhD in medical biotechnology from Medical University of Graz, Austria in 2013. He moved for post-graduation postdoc research from Internal Medicine, University of Iowa, USA, and currently working as assistant research professor in the same institute. He has 21 publications for his credit in reputed national and international journals, review articles, as well as book chapters. He guided many PhD and MD research scholars. He has 2 major research projects which are under progress. Sir has received 7 awards of recognition for his dedication towards research. To name a few: Sanofi Aventis Award for Outstanding Research in 2012, Young Investigator Award from International Atherosclerosis Society in 2018. He is an editorial board member for the Journal of Advanced Health Informatics and the Journal of Drug Delivery and Therapeutics. He is also a reviewer of ad hoc journals, and he is also a member for various academic committees too. His contributions are unlimited and it's are outstanding. So we are really glad to have an opportunity to listen to such a great person today. So without delay, I want our participants are eagerly waiting to listen to you. So let us start with the session. So I now request Dr. Prakash Dodpatar to kindly take over and begin the session. Thank you.
So, can you see my screen now?
Yeah.
Okay. First of all, I would like to thank Indian Academy Degree College and Dilshad and all the faculty members for this opportunity to talk to all of you with the main focus on students. And I really try to motivate and inspire as much as I can. And Dilshad has been my very good friend from degree college, and she has been very competitive since then, and she really tried to pull me to this talk from almost more than a month. She has been trying, and I'm grateful for that. So today I'm going to talk mainly on the cardiovascular interventions. It's just a very broad area actually. So to be very specific, I'm going to talk more on like immune cells in cardiovascular diseases. So what we are going to discuss today, so cardiovascular diseases with special emphasis on heart attack and stroke. And then we will also briefly go through the underlying cause of cardiovascular disease, which is the underlying, which is the buildup of plaque development called as atherosclerosis, and how immune system plays role with focus on macrophages during the development of atherosclerotic plaques. And in addition to that, how metabolic adaptations of macrophages within the plaques resulting from different types of like inflammatory and anti-inflammatory phenotypic cell types. And how glycolysis, which is a metabolic state, one of the cycles in which you in masters and undergrad you study how glycolysis plays role in shaping macrophage phenotype, and how targeting metabolic enzyme genetically improves atherosclerosis outcome in mice. So what are these cardiovascular diseases? So these are the group of diseases actually. So they mainly compose like heart and blood vessels. So when it comes to heart, it is called coronary heart disease, where there is a dysfunction in blood vessels supplying to the heart muscle. And when it comes to brain, this is called cerebrovascular disease. And when it is a periphery, like arms and legs, this is called peripheral arterial disease. And less known ones are the rheumatic heart disease, where damage to the heart muscle takes place and heart walls as well from rheumatic fever, mainly caused by streptococcal bacteria. As well as less known ones are congenital heart disease, where malformation of heart exists in their structure right from the birth. And in addition to that, so even a DVT, otherwise called as deep vein thrombosis and pulmonary embolism, where blood clots in the legs and leg veins that dislodge and move to the heart and lungs via circulation. So these are the group of cardiovascular diseases. Cardiovascular is not just one disease. So how this is the distribution of cardiovascular disease deaths are now split in male and females. So on the left hand side, we have male data. As you can see here in the red colored graph, it's a ischemic heart disease, which is nothing but heart attack. Simple word is heart attack. Ischemic heart disease means heart attack. So if you can see, like almost 50%, like 46% in males. So cardiovascular disease deaths are due to mainly heart attack. And again, next contribution is from the cerebrovascular diseases, mainly stroke. And remaining ones, like small contribution is by other inflammatory heart diseases like 2%, and other cardiovascular diseases by 11%, and rheumatic heart disease by 1%, and hypertensive heart also contributes definitely by 6%. And if you see, I cannot say completely in contrast to male data, in females, though ischemic heart disease contributes to the overall cardiovascular deaths, so still in female data, we have like more of cerebrovascular diseases. And cerebrovascular as well as ischemic heart, they are almost equal, you can see, but lesser than men, like especially heart diseases. And vascular diseases, they are more in female as compared to males. And small contribution is by inflammatory heart diseases like 2%, and other rheumatic heart disease by 1%, hypertensive heart disease by 7%. So where are we standing? Where India stands as compared to the USA or global data? So there was a very recent paper in one of the amazing journals called JACC. So as you can see on the left hand side, we have like a CVD. On the y-axis, we have cardiovascular deaths daily. Daily stands for disability adjusted life year, which is the mainly summary measure of population health that accounts for both mortality and non-mortality health consequences. If you see if you see the bar graph now, so we have a blue color, which is a global data, and it's not clearly red though, it's India, where gray color stands for United States. So this is the 90s data, 2000 data, we have like 2010 as well as 2016 data. So if you see carefully on the leftmost side, in the 1990s, where India was standing in the center bar is like India, like tobacco usage, though it was more, but it was compared to other countries, it was almost equal, and definitely all countries were having low physical activity. But the dietary risks even then we're still higher, but as compared to USA and global data, like a blue, if you compare with the blue bar. But if you come to the rightmost side, where it's we have a recent most data, which is 2016 data, we have like tobacco usage, it is increased compared to USA data, and as well as low physical activity is also increased slightly. So this is also not a good sign for developing countries like India. And if you see dietary risks, so overall globally it is decreased. I mean, compared to India, I can say globally it is decreased, or in the USA it is even more better now. I mean, it's far better. But India, where are we standing compared to global data? We have a dietary risk factors that are already increased a lot and contributing to cardiovascular deaths globally. So how this is going to impact economically? So on the left-hand side, we have the overall dollar amounts in billions, and on the x-axis we have years. So we are now in 2020. So if you go like in couple of years, like in 2023, so the expected, if the current status goes unchanged, like if you don't do any improvement, if you don't take proper measures, so direct cost of cardiovascular disease treatment in the USA, just in one country, it is expected to be nearly $190 billion. So the purple color graph shows. So if you come in the curve of green color, if we take proper measures, we can definitely bring down the total cost to nearly like $100 billion. So almost, from 200 to 100 billion, we can still control if we take proper measures. So what are those measures? How it is going to affect? What is the mechanism? I'm going to take you through those information in coming slides. So now coming to what are the key facts here? As we know, cardiovascular diseases, we know the types. So now these diseases are the #1 cause of death globally, which we already checked. So which means more people are dying annually from cardiovascular disease than any form of any other cause. So this is resulting in an estimated almost 18 million people died from CVD in 2016, which is really a lot, representing 31% of all global deaths. You can see the impact. Out of these deaths, 85% are due to heart attack and stroke, which is cardiovascular and cerebrovascular related deaths. Over 3/4 of cardiovascular deaths takes place in low and middle income countries. So this is again one of the important things we can check here. So out of the 17 million premature deaths in this context we call deaths that occur below the age of 70 due to non-communicable disease in 2015. 82% are again in low and middle income countries. So this is again a very important concern. And 37% are caused mainly by CVDs. So how they are contributing? So most of the CVDs can be actually prevented by addressing just behavioral risk factors. What are those? Like tobacco use, unhealthy diets, obesity, physical inactivity, and harmful use of alcohol, using population-wide strategies. So if you address these concerns, so we can really affect the current growth of this disease. And people with cardiovascular disease or who are at high risk of CVD, which due to these factors like hypertension, diabetes, hyperlipidemia, or already established diseases, they need early detection and management using counseling and medicines as appropriate, which is really hard in low income and middle income countries. So what happens now? How do we know what are the symptoms of cardiovascular diseases? So often we can say there are no symptoms for the underlying disease of blood vessels. Actually, in general, like heart attack or stroke may be the first warning sign of underlying disease. So what happens when there is a heart attack? There are symptoms for that. So like pain or discomfort in the center of the chest, pain or discomfort in the arms or the left shoulder, elbows, jaw, or back. And what about the stroke? So most common symptom in case of stroke is sudden weakness of the face, arm, or leg, most often on one side of the body. So how Americans are affected by these cardiovascular diseases. So this is just to give you very vague background. So nearly 46% of Americans are estimated to have hypertension. So almost 116.4 million US adults. So these are the findings from recent data which was taken in 2017. So if you see the middle of the slide, it means 1 in 6 males and 1 in 7 females in the USA are current smokers. This is again very bad sign, and it is estimated that like in coming to 15 years, more than 130 million adults, which is almost 50%, like 45% of the US population is projected to have some form of cardiovascular disease. And this is going to cost at least $1.1 trillion by 2035, with direct medical costs projected to reach 748, almost $750 billion. So this is again very bad sign. But how many people are aware? What are the, like only 22.5%, like 1 in 5 adults in America on an average, they are reported to have like adequate leisure time for aerobic and muscle strengthening activities to meet physical activity guidelines. Not all, you can see only 22% are just they are aware of this, and they have time. I mean, no, I cannot say that time, but they are aware to understand the impact of this, and they are really taking care of their body. So this is by recent American Heart Association data. So what we understood so far, we have understood the facts and figures of cardiovascular diseases globally, and now let's focus on the immune system in cardiovascular development. So what is immune system? I know that most of you, I mean all of you know that immune system is a wonderful collaboration between cells and the proteins that work together to provide defense against infection. So where it starts? So immune system, immune cell system, or any cells in the body for that matter in this case. So they develop from the bone marrow. So I'm not going to talk about immune system chapter or something, because this is really wide. What I can very briefly take you through, our cells of concern, how they are generated, why they are important. So as you all know, like from bone marrow, we have like hematopoietic stem cells, which will give rise to either myeloid or progenitor cell types. So again, we are not going to talk about lymphoid progenitor cells. We are going to talk about myeloid progenitor cells. So among those important ones in case of cardiovascular disease are neutrophils and macrophages, because these are the cells that are deposited or initiate atherosclerosis or buildup of plaques in the arteries. So what happens if you see on the left side, in healthy person or in healthy tissue or in healthy condition, bone marrow actually releases just limited number of cells, and those cells are they migrate into their destinies like tissues, and there are very minimal number of immune cells everywhere they are present. If you see now, this is the heart on the left hand. If you see, this is the healthy heart, where we see hardly any macrophages here, for example. So now if you see the artery, this is a very clean artery. There are few of the macrophages or cells, but definitely they are very, very less in number. So what happens towards the right side of the slide, if you see, so over a period of time, like when there is a disease, like for example, it can be diabetes, old age, obesity, psychological stress, increased cholesterol or LDL cholesterol, that for that matter, inflammatory cytokines, growth factors, danger signals like TLR4 ligands, like you can say like bacterial infections and all, like, and also inflammatory comorbidities. So what these conditions they result into? They result into increased bone marrow production, so which is also called as hematopoiesis. So then that results in the clonal hematopoiesis. So particular clones of cells are released into the circulation, into the destiny organs. So this will also result in the extramedullary hematopoiesis, also results in the myeloid bias and shift towards production of inflammatory cell types. So though bone marrow produces all sort of cells, so here in disease condition, this starts giving signal for the production of inflammatory cell phenotypes. So what they do, so they enter into the tissues, as you can see here, just below on the right side only. I don't know how can I get the pointer here. Okay. Okay. Yeah, you can see the pointer here. If you see here, there's so many immune cells as well as fibroblasts and also cytokine release and also neo vessel formation in the disease condition. If you see on the right side, here is the artery. If you see here over the period of time, so when there is a plaque progression, this is the intimal layer. There is a plaque formation. I will explain this, how these plaques are formed, everything in very detail in coming slides. So overall, what I want to show here is that when there is a disease condition, there is more recruitment of immune cells in the damaged area. So what it does, so overall, it results in the increased number of leukocytes in the tissues, increased proliferation, increased inflammation, and also shift towards macrophage subsets, which is like from anti-inflammatory to pro-inflammatory. And also there is a increased fibrosis as well as negligence of steady state tasks. This is the condition which is called as atherosclerosis, when there is a plaque formation in the artery. So now we will, in detail, we will go through the what is this disease and how immune system is playing a role. So now in the coming slides, I'm going to take you through what is atherosclerosis, how cholesterol deposition and inflammation affect the process of atherosclerosis. So what is atherosclerosis? It's a disease of cardiovascular system affecting the vessel walls. So it leads to the narrowing of arteries or complete blockage. And main components are endothelial dysfunction, lipid deposition, inflammatory reaction to the vascular wall, and also that ultimately resulting in the remodeling of the vessel wall. How this is happening, I will show you. So this is a wonderful slide. I can tell you the process of atherosgenesis or atherosclerosis, how this takes place. So if you see, this is a very healthy, normal artery. So this is a section. If you see here, this healthy artery has like a 3 parts, and outermost layer is called externa or adventitial layer, and the middle layer is called as it's a media or medial layer, and innermost layer is called intima, that is lined by endothelium. And in the medial layer, you can see in a normal artery, you can see smooth muscle cells that are present. So what happens in the early atheroma or early atherosclerotic plaque progression? So there will be deposition of lipid particles here in the intimal layer. If you see here, very less of plaque is, I mean, it's early stage, though there is less plaque here, and there is a thin cap here. You can see this is the cap of smooth muscle cells. So what happens if this situation is left untreated? If you don't do a good exercise, or if you don't take proper medication, or if you just ignore this, so this results in the, over the period of time, more and more lipid or fat, I can tell you, fat is deposited into the plaque, and this results in the thinner fibrous cap. So when this becomes thin, it becomes vulnerable. That's why it is called as vulnerable plaque. So there is a large lipid pool, and there will be a lot of immune cells or inflammatory cell migration into this intimal layer. So now, what happens next? If you treat it, you can stabilize this. It cannot become zero, but you can stabilize this. So what happens if you stabilize this? There will be small lipid pool here, you see here, and there will be thick fibrous cap, which actually prevents this area from damage. And then this overall thick plaque preserves the lumen area. So even though this lumen area is narrowed here compared to this or the healthy one, so here it is getting better. So what happens if you don't take even any precautions or any medication at this stage? So this results in the formation of thrombus or thrombosis of ruptured plaque. What happens? Here the fibrous cap becomes thin, and that results in the breakage, and this results in the clot formation. So what happens now? This is nothing but myocardial infarction, otherwise called as heart attack. So this is the heart. If you see the left coronary artery, this is the right coronary artery. So in the left coronary artery, when there is a plaque rupture and clot formation, so this is called as heart attack. That's why many people, when they say heart attack, this is the region that is actually affected. And if you actually treat or heal this stage, at this stage, there will be very narrow lumen, formation though, but blood flow will be more, but there is at least a stable plaque. So it is still not a good sign, but at least this is not like broken or damaged an artery. So what happens next? So if you go through even little deeper, as I initially said, there are macrophages, cholesterol particles that play a role here. So what happens? These are the below, you have see the steps: endothelial dysfunction, inflammation, oxidation, plaque instability. So what happens in each stage, I'll tell you. So there are molecules generally called as irritants. They are called, irritants can be like increased cholesterol in the circulation, smoke particles, increased blood pressure. So why they are called as irritants? They act as they irritate the endothelium. Endothelial is the barrier actually. So when there are more of irritants in the circulation. So like in monocytes, like LDL cholesterol or low density lipoprotein cholesterol or hypertension or smoke particles, they first damage the endothelial layer. So endothelial cell damage, when this takes place in the first stage, so they attract monocytes. So why they are called monocytes? These monocytes, when they are in the circulation, they are called monocytes. Once they enter into any tissue for that matter, now they are called as macrophages. So damaged endothelium, which express adhesion molecules, as the name suggests, they adhere, they attract these monocytes. So monocytes bind to this and they enter into the intimal layer. So once they are into the intimal layer, so there comes also this cholesterol particles, LDL cholesterol, they also enter into the damaged endothelium. It's like there is a damage, they enter. It's very simple concept. And once they enter, these macrophages, they take up these low density lipoprotein particles. But in this area, when there is more of oxidation, these low density lipoprotein particles are modified. They are oxidized. Now they are called as oxidized LDL. So these modified or oxidized LDL particles are taken up by macrophages in the intima, and then macrophages become fatty cells, like fat sort of cell. They are called as now foam cells. So why they are called foamy? Because they appear like foam. So they have a lot of number of lipid droplets. They store all these lipids in their lipid droplets. So what happens over the period of time? Over the period of time, so there will be oxidized environment in this area, in the intimal layer. So these foam cells, and they start releasing number of inflammatory cytokines and matrix metalloproteinases. They all damage the smooth muscle cell thick layer, which was thick. If you go back to previous slides, now that results in the plaque rupture. So this rupture, once this is ruptured, so they release their contents into the circulation, they form the clot. That's why this is called as plaque instability or unstable plaque. This is called unstable, and this results in the thrombus formation. And now, how, even if you go one step even deeper, how this inflammation is playing a role here. On the upper side, we have the arterial lumen, and the bottom side, we have like arterial intima. So if you go back to the intimal layer, which I explained in the first slide. So if you see, when there are blood monocytes, when there is an endothelial damage, you see in the green, so they are the adhesion molecules expressed by the endothelium. So they attract, like for example, VCAM1, there are also like ICAM1. So they attract monocytes, and monocytes are adhered to this epithelium, and they are now, they migrate into the intima. You see the gap here, there is a damage, endothelial damage, and that is where monocytes enter. Now they become macrophages. What happens to this? Now they start expressing number of receptors, like CCR2 that specifically binds to MCP1. And once these monocytes become macrophages, they start expressing another sort of receptors called as scavenger receptors. As the name suggests, scavenging means taking up. So once they start expressing scavenger receptor, they take modified LDLs. So they become foam cells. Now they are called macrophage foam cells. So which are rich in lipid droplet particles. And these macrophage foam cells, they are not good if they are left there for a long time. So what they do, they actually release ROS They release cytokines, they release matrix proteases, tissue factors. Overall, all together, they contribute to the plaque rupture or unstable plaque. And over the period of time, even these cells undergo apoptosis as well as necrosis. Now, so far what we have studied, we understood a little bit of facts and a little bit of immune cell function, and what we understood about the immune system inflammation during different stages of diseases and disease progression. Now let's talk about the role of lipids and fats in disease progression. So before again I explain how lipids are playing a role in plaque progression, so I will briefly tell you about how lipid metabolism pathways are playing. I'm not talking about very deep metabolic pathways. I just want to take you very lightly through overall how fat is absorbed by cells and how our machinery, body natural mechanism plays a role in clearing of these fats. So there are like basically two pathways, like exogenous pathway and endogenous pathway. So what happens in exogenous pathway? So when there is a dietary fat, so when we eat fatty food, or general any food, so fat from that food actually is first absorbed into intestine, and intestine releases those fatty particles in the form of chylomicrons. So these are large lipid-rich particles. These are the biggest ones. And once they enter into the circulation, they are acted upon by lipases called as lipoprotein lipases or LPL in the capillary. So when these lipases act on chylomicrons, they break down triglycerides. So that releases free fatty acid. So some free fatty acid they go and get deposited in adipose tissue and muscles for normal activity. And now once they lose more of fat, now they are called as chylomicron remnants or simply remnants. So these remnants are taken up now by the liver. Now starts the endogenous pathway. What happens now? We will see. This is very interesting. So now these remnants, they enter into the liver, and now liver releases another type of lipoprotein particles called as VLDL, or very low density lipoprotein particle. So these VLDLs, they are again released into the circulation by liver, and these are again acted upon by enzymes called as lipoprotein lipases, simply lipase, and they break down further of triglycerides. And these VLDL particles have more of triglycerides, less of cholesterol. And when these lipases act on the triglycerides, they lose more and more of triglycerides, and those free fatty acids that are broken down, again they go to the adipose tissue and muscle. And the remaining one, they still stay in the same particle. Now those same particle, after they lose more of fat, they are called as intermediate low density lipoprotein particles, or simply IDL. So these IDLs either directly taken up by liver again, or these become low density lipoprotein particles. So these IDL are simply from VLDL to LDL if you consider. So LDL particle already lost almost all of its triglyceride, and it has now still rich in cholesterol. So that's why LDL cholesterol is called as bad cholesterol. Whenever doctor tells you to check your blood parameters, so they check for the LDL cholesterol. So when there is more of LDL, so this LDL is very dangerous. So what happens? This LDL either goes to the liver via LDL receptors, and it also gets deposited in extrahepatic tissue like arteries. If you go back to my previous slides, when I was saying LDL cholesterol, and that was this cholesterol that was getting deposited into the arteries. So what happens there? So in the arteries, when LDL cholesterol is entered, macrophages, which play important role in clearing them, they become foam cells, right? So at this extrahepatic tissue is now acted upon by HDL, which is also called as good cholesterol. So HDL level should always be higher than, definitely higher, and LDL cholesterol should be lower than the expected range. Like LDL is, I think, 180 to 200 mg per deciliter, whereas HDL generally it should be above 50, but generally most of us Indian population has around between 40 and 50 mg per deciliter. So what HDL does in the circulation, HDL actually takes up LDL cholesterol from the extrahepatic tissue and brings it back to the liver in a process called as reverse cholesterol transport. So HDL is playing a role in clearing LDL cholesterol from the arteries or extrahepatic tissues. So I don't want to really spend time on this particular slide because this is the classification of lipoproteins, where you can see biggest ones are called as chylomicrons, and then comes VLDLs, and then comes intermediate density lipoproteins, and then comes low density lipoprotein particles, which are very, very, very dangerous. And then comes high density lipoprotein particles. They are high density. They are not because they are called as high density because they are bigger. They are more in number though, but they are there's the smallest lipoprotein particles. So now we are going to talk on role of macrophages with special emphasis on its metabolic states in modulating inflammation during disease progression. Yeah, so macrophages. If you recollect initial what I was telling about the inflammation or anti-inflammation, how these cells they change their phenotype. So normally naive macrophages are called M0 macrophages, but when there are intrinsic triggers like intracellular pathogens, cytokines, lipoproteins in the circulation, or in any disease condition, these macrophages become M1 macrophages. M1 macrophages are the inflammatory ones. So when their number is increased, then there is more inflammation in the body. So these macrophages express more of ROS, they express other receptors. I will not go through the CD86, CD40, but in general, I'm telling you, these are called as M1 macrophages, which are bad macrophages or inflammatory macrophages. They release number of cytokines like IL-12, TNF-alpha, IL-6, IL-1 beta, MCP1, resulting in the pro-inflammatory microbicidal pathogen killing, as well as tissue injury conditions. But when there are intrinsic triggers such as immune competent complement apoptotic cells or cytokines like IL-4, which are anti-inflammatory, TGF-beta, IL-10, so these naive macrophages become M2 macrophages. These M2 macrophages are called as anti-inflammatory. These are they are pro-resolving macrophages. So they express markers like 206. This is also called as MR, and they also express CD163, CD36, and ultimately these M2 macrophages, they release more of TGF beta, IL-10, MCP1 low, TNF-alpha low, IL-6. Why it is written TNF-alpha low? They do secrete, but they are very, very, very lesser as compared to M1 macrophages. But anti-inflammatory phenotypes, anti-inflammatory macrophages, they clear apoptotic cells or dead cells in the intima. They have high phagocytic activity, and also they are wound healing. So how metabolic polarization of macrophages is taking place? So I said here there is macrophages behave differently when there is metabolic when there is change of depending on the triggers that trigger them, like towards M1 and M2. So what happens next? They actually, these intrinsic factors, they mainly trigger, they change their M1 and M2 phenotype depending on the their metabolic state. So what happens here is a metabolic polarization of macrophage. So this is the recently people are very much interested in this particular type of studies. As you can see, when there is like LPS or interferon gamma treatment in macrophages, so cells become they start expressing less of mitochondrial activity, less of oxidative phosphorylation that results in the inflammatory phenotype. But if you treat the cells with IL-4, for example, of course, don't look at this part, but in general, when you treat cells with IL-4, they express increased mitochondrial activity as well as normal oxidative phosphorylation. And these macrophages are called as anti-inflammatory phenotype. So why it is very important to understand metabolic state? So it's all about metabolism, metabolic activity of the cells that gets affected, resulting in the altered inflammatory phenotype of cell system. And how glycolysis is playing a role here, because glycolysis is the basic step in any metabolism. So as if you can see here, when glucose is converted to pyruvate, so what happens in the normal cycle, normal respiration cycle? So there are the intermediate cycles, but this result in normal oxidative phosphorylation results in the 32 molecules of ATP molecules. So when there is aerobic glycolysis, especially in case of cancer or any inflammatory condition, there will be more of lactic acid production. So this is also called Warburg effect. So where only 2 molecules of ATPs are produced. So why it is important to know briefly. So my research area of research is on the metabolic state of the cell, which I'm going to explain in coming slides. So on the left side, if you focus on the macrophages, so resting macrophages, they do express TLR4. They have normal glycolysis. They have enzyme like PKM1. In this case, this is called pyruvate kinase M1. So that converts that breaks down the final step of glycolysis and promotes normal oxidative phosphorylation. When cells are activated, like in case of disease condition, so when like in this case, like when cells are treated with LPS, for example, TLR4 is activated, there will be more of glycolysis. So when there is more of glycolysis, as I said, the enzyme pyruvate kinase, so which plays in the breakdown of glucose into pyruvate, so there will be more of dimeric forms of this PKM2 that enter into the nucleus and binds to the HIF alpha, HIF1 alpha and beta subunit, and brings about inflammation by releasing more of cytokine like IL-1 beta. And when in this case there will be less of beta oxidation, so that affects the succinate formation. So succinate binds to HIF alpha and stabilizes the HIF alpha binding to the DNA in the nucleus. So when cells are, this is case when cells are inflammatory or pro-inflammatory. If the cells are anti-inflammatory, for example, so when they display normal glycolysis, but the PKM2 gene, which is in dimeric condition, in inflammatory condition, so in normal condition or in anti-inflammatory condition, it is in tetrameric form. So when it is in tetrameric form, it doesn't enter into the nucleus, and thereby not releasing inflammatory cytokines. So here in this case, when the cells are anti-inflammatory, when they are good, so there will be normal oxidative phosphorylation and more expression of anti-inflammatory cytokine like IL-10. So what I've been doing in recent days, I'm checking the role of myeloid specific PKM2 or pyruvate kinase enzyme role in atherosclerosis. So myeloid specific means I'm checking the role of PKM2 especially from macrophages. Okay, and now just to give a very brief background as to why I am using PKM2 or why this gene. So because abnormal glucose metabolism and enhanced oxidative stress increases or accelerates cardiovascular disease, and glucose metabolism in lesion residing macrophages is upregulated, as well as monocytes from cardiovascular disease or coronary artery disease patients are very much prone to differentiate into IL-6 and IL-1 producing effector cells. Means these monocytes from the patients are more of inflammatory cells or M1 sort of cells. So this hyperinflammatory state is mechanistically dependent on glycolytic activity and glucose-induced ROS production by the mitochondria. So whatever the brief background I was giving you before, now if you try to connect the dot, you will understand why this particular enzyme is important and why I am studying this here. So now ROS production within the macrophages changes the oligomeric assembly, like from dimer to tetramer. I will explain that even better in coming slides. So oligomeric assembly of the redox sensing enzyme, which is PKM2, enabling its nuclear import and phosphorylation of STAT3 to promote IL-6 and IL-1 beta production. So this PKM2 enzyme, it's a key metabolic regulator and is widely studied in aerobic glycolysis of tumors. So in cancer, it's very well studied, but in cardiovascular system, it is recently, the point of focus. So mammalian cells contain mainly two genes, pyruvate kinase genes. One encodes for PKL, PKR in the liver, and RBCs respectively. The second gene encodes PKM1, PKM2 by alternative splicing of PKM3 mRNA. What happens? Last step of glycolysis is mediated by PKM1. PKM2, on the other hand, upregulated in tumors and exists primarily as enzymatically inactive monomer or dimer that enters the nucleus. So now PKM2, actually you can say in case of tumor or in case of inflammatory condition, PKM2 expression is increased, and that enters into the nucleus of the cell and brings about more of inflammatory cytokine production. So in my work, what I studied, first I checked the expression of PKM2 in human and mouse tissue. These are on the left hand side, we have immunofluorescent images of human artery as well as mouse aortic sinus, where plaque progression can be very well studied. So on the green color, we have like PKM2 gene, and in red color, we have CD68, which is macrophage. First, we checked PKM2 is expressed in human diseased arteries or not. So yes, we did find PKM2 expression, and then we also checked the co-localization with macrophages by CD68 co-staining. We found the yellow ones are the co-localized expression where PKM2 is present in macrophages. So this confirms, yes, PKM2 is present in other cells, but yes, it's also highly expressed in macrophages. Similar observations we found in mouse tissues. And then what we did, we generated macrophage specific or myeloid specific PKM2 knockout gene. So as you can see on the right hand side, we characterized the mice by genotyping. On the left hand side is the PKM2 floxed mice, where PKM2 gene is present. On the right hand side, we have lysine cray. Lysine cray is specifically, lysine is expressed in myeloid cells. Cray positive or plus or minus, it means when the cray is present, this cleaves the floxed sites. So it means again there is no PKM2 in myeloid cells. So this is a control called as wild type, just to compare with. And then we also check the protein expression level just to make sure, yes, we have successfully knocked out PKM2 in macrophages. So on the left hand side we have control, which is lysine cray negative. On the right hand side we have lysine cray positive. When this gene is present, it cleaves PKM2. So that's why we don't see expression of PKM2. And on the right hand side we have the quantification. So how we designed the experiment? So we used PKM2 floxed mice and PKM2 lysine cray bone marrow and transplanted the bone marrow into atherosclerotic prone atherosclerotic prone mouse model called as LDLR knockout mice. And we fed mice with a Western type diet, which is high fat, high cholesterol diet actually, for 14 weeks. And we studied Atherosclerosis. So now coming to the data. So myeloid specific deletion of PKM2 decreases atherosclerosis. So we used both male and female mice, and we found on the right hand side, in the left hand side we have iota of mice, and on the left, upper panel is the female data and the lower panel is the male data. We have the left hand again, we have, this is oil redo stained tissue samples. Oil redo specifically stains the fat. So more the red color, it means more the fat. So as you can see here, compared to controls in knockout mice, we found significantly reduced plaque formation. And here is the quantification of the data, same data. So we found significantly reduced plaque formation in knockout mice as compared to controls. Similar observations are also found in male mice, which is the bottom panel. And then we studied the plaque progression in aortic sinus. So this is also called as aortic root, in the heart root. So when we studied, like we stained with the Verhoff von Giesen, so this is overall stains, overall plaque in the aortic root. This is the plaque area, if you see here, and this is the plaque area. So, and if you see in the knockout mice, we found there is significantly less plaque here compared to these mice. So upper panel is again the female mice, and the lower panel is the male mice. So we found on the rightmost side, we have the quantification of the data. We found significantly reduced plaque progression in PKM2 knockout mice as compared to control mice. The similar observations are also seen in male mice. And then we wanted to understand why there is a less plaque formation. So we stained for macrophages. So initially, again, if you go back to my introductory slides and the backgrounds, so how macrophages are playing role. So when PKM2 is deleted in macrophages in mice, so we found this is the black color is the immunostaining for macrophages. In knockout, we found less macrophage accumulation or migration as compared to controls. Similarly, in male mice, we found here significantly less plaque as well as less macrophages compared to controls. So rightmost side we have the quantification. It says MAC3 positive. MAC3 is the antibody that specifically stains macrophages. So we found significantly less macrophage positive area both in female and male knockout mice as compared to controls. However, as I was also talking about the lipids that play a role in plaque formation, so we did not see any difference in the cholesterol level or in triglyceride level, both in male and female mice groups. So basically it means it's the local inflammation in the aorta or the local inflammation in the heart that is playing role in the plaque progression, not the level of cholesterol in the circulation. So the take-home message of the overall talk is that the risk of atherosclerotic event can be decreased by cholesterol low diet, increased exercise, smoking cessation, control of high pressure, drugs like statins, fibrates, estimab, antioxidants, and increased glycolytic activity results in the inflammatory macrophage phenotype, where PKM2 is a glycolytic enzyme which is playing a role in inflammation. So targeting myeloid-specific PKM2 gene results in the amelioration of atherosclerotic lesion progression without affecting plasma lipid levels. So with this, I would like to thank my collaborators and my colleagues, Anil Chavan, Stephen Lenz, Isabella, my colleagues Nirav, Manasa, Manish, Rishabh, and Madan, who are recently, half of them recently joined and they are working with us. So I would like to thank all of them. So with this, I'm finishing my talk, and if you have any questions, I'm very much happy to answer. So please go ahead.
Thank you, sir. Thank you, Prakash, sir. Now the session is open for discussion. You please put your questions in the chat box. You're not supposed to ask. So we are going to read it to the speakers. Any questions? Yes.
So can I type all the answers if I have all the answers in this chat box?
No, sir. Wait, sir. Sir, there is a question. I'll read for you. What is the difference between stroke and massive cardiac arrest?
Well, stroke and cardiac arrest, they're completely different, first of all. So how? Stroke is like a attack of brain, where the clots from the plaque rupture, they move to the brain and they block the blood supply to the heart. This is called stroke. And the cardiac arrest is completely different. I think you wanted to ask about Mr. Vasant Kumar. Maybe you wanted to ask about myocardial, exactly, heart attack or myocardial infarction. That is true. Heart attack is where the blockage happens in the heart. Stroke is blockage happens in the brain. So this is the difference. And cardiac arrest is completely different. If you see, recently there are many actors are dying, the many prominent figures by cardiac arrest. Cardiac arrest is like a electrical impulse that is affected in the heart. So which is very bad, very dangerous. Cardiac arrest is very dangerous than heart attack or myocardial infarction. So all sudden deaths, massive cardiac. Yes. Okay, what would be the possible reason for silent attack when a person doesn't have any cardiac history? Yes, that's what I'm saying. This can be hereditary. We don't know. Sometimes the type of drugs we take. Sometimes some people with just by alcohol they have palpitation of the heart. Some people with coffee they have palpitation of the heart. With salt they have palpitation of the heart. There is not one reason for this. Very recently there was a very young actor in Kannada industry. He died of cardiac arrest. And today, the choreographer in Bollywood industry, she died of cardiac arrest. Why? Because Definitely, age is one of the factors, but even young ones between the age of 30 and 40, they die because of cardiac
Come
arrest.
first.
Sometimes people who are taking some type of restricted
जल्दी आ
drugs,
जाओ मैम.
so they may also face this
ऑफ
issue.
है जल्दी आ जाओ.
There is a electrical impulse that is badly affected in the heart, or suddenly when it stops, that stops the functioning of the heart. When heart function stops, blood pumping stops. When blood pumping stops, blood supply to the brain stops. When blood supply to the brain stops, brain function stops. When brain function stops, whole body function stops. So this is very nicely connected to each other. That's why cardiac arrest is very much dangerous than heart attack. Heart attack, definitely it's just narrowing of the artery. Definitely you know the sign like chest pain, heart pain. If you do exercise, you can still prevent this. But with cardiac arrest, it's very dangerous. That's why within 10 to 20 minutes before our brain, once it doesn't get any blood supply or oxygen supply, so once it stops functioning, it has to be very quickly, heart has to be like activated or made functional. Otherwise, brain cannot function. So when there is lack of oxygen supply, heart dies. Oh, sorry, brain dies. So when brain dies, so whole body is gone. Then what is your question? What could be the probable reason for hereditary related with? Of course, there are hereditary issues. Like for example, hypercholesterolemia, genetic hypercholesterolemia, so hypertriglyceridemia. So there are hereditary issues also, which I already explained in the introductory slides.
Okay. So there is one question. What is the? There is one more question. So why heart cancers are less frequent?
I don't know about cancers. Heart cancers, I don't know, because that is not something common, and definitely we never study heart cancers, so we don't know about heart cancers. And there is another question: what is the correlation between
glucose and plaque formation?
Yeah, completely. Glucose is definitely, it's just glucose level in the blood. Definitely that affects, because whenever there is more glucose, ultimately everything is converted back into fat. You can, for that matter. So, and definitely diabetes in this case, it's just a comorbid condition. When there is more glucose in the circulation, that results in the inflammatory overall environment in the body. So when there is more of inflammation, as I said again, so that contributes hugely to all diseases, including cardiovascular diseases. So more glucose is very dangerous. I can tell you only one thing: glucose is actually a toxic poison. It's a slow poison. So that's why if you avoid this as much as you can, it's always the best thing. So glucose is in fact more dangerous than fat, for that matter, just for your information.
Okay.
Okay, then what else? I hope I answered.
Yes, sir. You have answered almost all the questions, I think.
While my colleague Soumitra presenting, I will still try to type your answer.
Sure.
So you can still type. Okay. Thank you so much.
Okay. So now, thank you so much, Dr. Prakash Dutpata, sir, for explaining us the process of
Prakash, because it's okay.
Okay. It's really very nice talk. It's all about role of immune cells, how it will affect the atherogenesis, and also we really got a very good conclusion about the topic and about your research, which is on myeloid pyruvate kinase. Really, we are very much thankful to you, sir. Thank you once again.
It's my pleasure.
Thank you. So, yes. Now let us start with the session 2. I request Ms. Pushpa Reddy, Department of Biochemistry, will continue with the session 2.
Thank you, Professor Dilshad. Good morning, everybody. I am Professor Pushpa Reddy from Department of Biochemistry, Indian Academy Degree College, Bangalore. I feel proud to introduce our outstanding alumni, Dr. Soumitra De Chaudhary, as a resource person for this international webinar, Trends in Biochemical Research. To tell you in brief, Dr. Soumitra obtained his master's in MSc Biochemistry from Indian Academy Degree College, Bangalore, and further on, he obtained his doctoral from Indian Institute of Science Education, Bhopal. Subsequently, he carried on his postdoctoral research at National Institute of Health, US. He has very good publications in PubMed. His career goals include understanding the molecular mechanisms of synaptic development and extend the knowledge to a mechanistic understanding of neurological disorders. His PhD training started with presynaptic studies, and later he continued with his postsynaptic studies research in postdoc research. Dr. Soumitra is currently working as a visiting postdoctoral fellow in National Institute of Health Center of on Compulsive Behaviors. Now, may I request Dr. Soumitra to present his knowledge on the topic Understanding Synapse Development using the Drosophila Neuromuscular Junction, an emerging concept in biochemical research. So it's over to Soumitra.
Okay. Thank you so much. Am I audible?
Yeah, you're
Okay.
audible.
So first of all, I'd like to thank Indian Academy Degree College for this invitation. And it's really an honor and privilege to be presenting in the same college from where I passed out. And what's really nice is the kind of work that I'm doing is partly also because of the motivation that I got during my master's. And if there are master students around, you probably a course in your semester called Molecular Physiology, and that was one course that fascinated me so much that I kind of tried to take that as my research study. And so quite a lot of the thing that I'll be showing you now involves a lot of signaling, which is something that I first came across during the molecular physiology classes. Um, Again, I'd like to thank the principal and the faculty of the Department of Biochemistry for this invitation, and I think it's an absolute honor to be presenting my work. So let's get started. All right. I think I have to share my screen. Okay. So I think my screen is visible.
Yes.
Okay. So all right. So yeah. So today what I'm going to do is talk to you about understanding synapse development using the Drosophila neuromuscular junction. And the image that you see here is that of Drosophila neuromuscular junction, and you see those rounded structures. That is where the nerve terminals are. It's called synaptic boutons. So I'll be coming to that in my later slides. But before we really get going, let me begin with something very simple. So I believe we've all seen a relay race. For that matter, some of us may have even participated in one. So the point of showing this slide or trying to tell you about relay race is that there is something which is really happening here, and that is uh, the baton is being passed from one person to the other, and I'm using the baton here as an analog for information exchange. Now, the question is, where in the body does this kind of information exchange actually happen? And undoubtedly, that happens in the brains, where you've got 100 billion neurons, and each of them forming like 1,000 synapses. And so total number of synapses would be 100 trillion. And each of these synapses work by exchanging information among themselves. So you can see here that there are two neurons, one passes the information and one receives it. So in every context, we talk of that as a presynaptic neuron, which passes the information, and that is received by a postsynaptic neuron. Now, I've told you that we can consider this as a baton that is being passed from one person to the other. Of course, the only difference is that instead of the baton that is passed from, say, the presynapse to the postsynapse, we have something called as synaptic vesicles, which are filled with chemicals called as neurotransmitters. And these neurotransmitters then are released in these minute 20 nanometer distance cleft called as the synaptic cleft. And then these neurotransmitters then bind to the receptors which are present in the postsynapse, and then the information or the signal is basically transduced downstream. Now, I've been mostly interested in understanding the different molecules that are present in the presynapse, or for that matter, even the postsynapse, and how these molecules interact with one another, how they formed, how they form modules which kind of help in the information exchange between the two neurons. And the way to kind of visualize these protein molecules or modules is that you can think that they are individually, they are kind of pieces of the puzzle. And when you put together this puzzle, you have a functional system. And then there is a lot of signaling that is also happening because of these modules. So I told you that this is what really happens in the brain, but then the question is, why should we really care? What is the need to really understand the synaptic processes or the information exchange, the synaptic transmission that is happening in the brain? Well, as it turns out, if some of these processes go wrong, you have disorders like schizophrenia and autism, and these are just to name a few. And so to study these, what I use is I use the fruit fly, that is a Drosophila melanogaster. And the advantage of using fruit flies is that they are pretty much similar to humans. Um, they have a brain, which is pretty much complex. They have something called as the ventral nerve cord, which is functionally equivalent to the spinal cord that we have. And they also have these segmental nerves, which we can then study. And also, flies are very easy to rear in the lab. They have a very short developmental cycle, and basically within a span of a week, you have an adult coming from an embryo. So this whole developmental cycle is pretty short, and that makes them a very suitable organism to work with in the lab. And to add to that, we've got a tremendous array of genetic tools available with us, with which we can perform all sorts of genetic manipulation and study the function of a particular gene. Uh, and recently, what has evolved is that we've got wonderful databases like uh, OMIM, that is the Online Mendelian Inheritance in Man. You have got the Undiagnosed Disease Network, UDN, and websites like Marvel. So these are the websites that coordinate or have data, patient data, and you can always look back at these particular databases, find the gene, find its homolog in flies, and then try to mutate those particular genes and see what is the phenotype that you can actually get. And what's even more important is that you can also find other fellow researchers who are working on similar proteins. You can also find clinicians who are working with patients. So these resources are of great importance because if you have a clinician who's working on a similar kind of a problem, you can always collaborate with the clinician and then, of course, extend your basic study in flies to the patient data and then understand the function of the gene in greater details. So, and also, I would like to take this opportunity here to do a little bit of publicity for this website, which is Droso for Schools. So if any of the students are really interested in understanding how the fruit flies are used in research and what are the similarities that they have with humans, and what are the resources that are available, the kind of genetics that one can do with flies, this website is really a great place to start with. And it's a basic thing, and I can understand a lot of things. So this is something that I'd just like to advocate. All right, so now coming to the model system that we use, that is the Drosophila neuromuscular junction. Um, the one in the top, the image is that of Drosophila larvae, but this is that of a first instar larvae. And then you have the blown up one. So this is the third instar. And you would appreciate that within a span of like 48 hours, the larva grows in size from the first instar to the third instar. And this growth is like 100 folds. So there is an increase in the muscle area 100 times. And to compensate or to coordinate with this 100-fold increase in the muscle area, you have the neurons also correspondingly growing and occupying or innervating these muscles. And since I said like this growth takes place in like 48 hours, so this is a wonderful model to study the developmental functions or developmental uh, to understand the function of a gene that could play an important part during development. And so the system that we use in the lab is that of a third instar larvae. So this is how a larvae looks like. And you can easily make this preparation of fillet by cutting along the dorsal line. So what happens when you cut this off is you kind of expose the larvae, you expose the ventral nerve cord. And then what is shown in the right is an immunohistochemical preparation. So you can basically open the larva and stain it for different markers, and then you're able to see the terminals. So and the other advantage of using Drosophila larvae is that you can appreciate that these segments are like arranged in a very stereotypical pattern. You have the same muscles on either side. So each side is called a hemisegment. They're divided into 7 different abdominal segments, A1 through A7. Um, and so you have the same set of muscles in the different segments, and you can always come back to the same muscle if you want to study an animal life. So so what is shown here is neuromuscular junction, that is the connection or the contact between a motor neuron and the muscle fibers. So you have the ventral nerve cord here. It is depicted in the cartoon. So you have the ventral ganglia here, and that's like the central nervous system. And you have the axons being projected from the ventral nerve cord, and they come out and have a contact with the muscle. And if you see in the bottom panel, you can see those rounded structures. So these are the synaptic boutons or the nerve terminals. So this is exactly uh, the business end of the nerve, and all the activity that I was talking to you about previously about the information exchange. This is the place where this actually happens. So if you kind of look in further detail, so now this is just one of those rounded structures that I showed you previously. And you see 2 different proteins. One labels the presynapse and the other the postsynapse. So the presynapse here is labeled with a protein that is called Brook pilot. It's an active zone protein. So that is at the presynaptic side. And you have something shown in the red that is the receptor, so in the postsynaptic side. So so this is a confocal image. And if you further go to the details that is shown here using electron microscopy, you can see the magenta here. That is the active, there's a presynapse with the active zone. The rounded structures that you see here are the synaptic vesicles. So they contain the neurotransmitters. And the dark structure that you see here, which is in the form of a T bar, is the active zone. And this is exactly what is shown in this image in the green. So this is the the part in the green. And this is a region where all these synaptic vesicles come, dock, and fuse, and they release the neurotransmitters. And when these neurotransmitters get released, they bind to these receptors, which are shown here in red. So they're again dense, and they're shown in red. And this is on the muscle side for the neuromuscular junction preparation. And you can see a very thin gap, and basically you can see two membrane structures. So this is the 20 nanometer gap or the synaptic cleft that I was talking about. So just to put things into perspective, what you see here in magenta is the presynapse. What you see here is rounded structures are the synaptic vesicles. This small gap is the synaptic cleft, and the green structure that you see here is the postsynapse. So now you've got a fair bit of an idea of how the Drosophila NMJ actually looks like. So with this now, I'll just get on with the main part of my talk. So I've kind of divided the talk into two parts. And what I'm going to do today is mostly discuss my PhD work that was done in IISER Bhopal. And the main, my thesis work was on AP2 adapter and endocytosis. So what I'm going to talk to you about is the synapse morphology and function, and what AP2 adapter does in clathrin-mediated endocytosis, and what happens if you deplete the AP2 adapter complex, or if you perturb the function of AP2 adapter. So what really happens to the neuromuscular junction? And the second part of this AP2 adapter and endocytosis, I'll be talking to you about the various signaling pathways that is affected when you have a mutant form of the complex. And towards the later part, I'll talk to you about a disease called amyotrophic lateral sclerosis, which is also called as the Lou Gehrig's disease, and a particular gene that is involved in ALS, that is senataxin. And our studies with the human form of senataxin uh, and our studies on like what the kind of results that we got expressing the different variants of this in Drosophila motor neurons, and what are the conclusions that we could draw. So as I said, the neurons are highly polarized. You have a cell body, you have a long axon, and then you have the synaptic terminal or the synaptic bouton. And now this is the business end of the nerve terminal, because all the vesicles are basically released into the synaptic cleft from this side, and then there are receptors here which take up the chemicals, and then of course the signal is processed downstream. Now, if you zoom in and try to understand what really happens here, is that you've got these synaptic vesicles which are filled with neurotransmitters, and when there is an action potential that comes in, you have calcium coming in through the calcium channels, and that leads to the fusion of these synaptic vesicles with the presynaptic plasma membrane, which is mediated by the snare complexes. So that leads to the release of these neurotransmitters into the synaptic cleft, which then get attached to the neurotransmitter receptors, and then of course the signal is processed downstream. Now there is a fundamental problem out here, that is, once these synaptic vesicles fuse, they essentially add to the membrane of the presynaptic space. So this is basically a bilayered membrane structure that comes and fuses with this membrane, and so you basically add this extra membrane. So if you don't recycle these membrane back, essentially what happens is that you're going to have a bulging membrane, and that will not further allow any more fusion, and a physiological consequence of that would be paralysis. So what are the ways by which you can basically recycle these membranous structures? So the way this happens is by endocytosis. So you have clathrin-mediated endocytosis taking place, and this is the clathrin coat that forms around these membranous structures. Then you have a GTPase called dynamine that comes and cuts this vesicle off, and then you have the vesicle being regenerated again. The clathrin coat comes off, and then of course you refill this vesicle with neurotransmitters, and this cycle continues. So this is basically called the synaptic vesicle cycle, and this cycle then actually continues. So one of the major modes of this mechanism of endocytosis is mediated by clathrin, and AP2, of course, is the adapter complex that mediates this process. but then there are other modes of endocytosis that also happens at the synapses, and I'll come back to that later. So, so when I started my PhD, I started with like this three major questions. So the first question was, what is the role of the AP2 complex in presynaptic vesicle recycling? So when you have to recycle these vesicles, what role does AP2 actually play? Then the next question was whether the AP2 complex plays an exclusive role only in vesicle recycling, or is it also required somewhere else? And the third question was, how is a defect in endocytosis linked to aberrant synaptic growth? So this question basically comes from the results that we got when we perturbed the function of AP2. So to tell you a bit more about the AP2 complex, so this has got four different subunits. The alpha and the beta are the two bigger subunits. You've got sigma 2, which is the medium subunit, and the smallest subunit here is the sigma 2. Uh, and this kind of holds together the whole complex. So what is interesting is that if you take away one of these subunits, the whole complex falls apart. Um, but there are studies in other model systems, particularly in the one in C. elegans, that shows that if you take apart this system, the alpha and the sigma can form a hemi-complex and can work together, and the beta and the sigma can actually form a hemi-complex and work together. So one of the things that we actually tested is whether that is true in Drosophila, but in Drosophila, this does not actually happen. So if you take out just one of these, everything else falls apart. So, uh, so what I then did was basically try to understand what the function of the sigma 2 complex was. I mean, sigma 2 in putting together the complex was. So when I started off, we had a mutant in the lab, and that mutant had a synaptic morphology, which I'll come to in the next couple of slides. And since the morphology pretty much resembled a bunch of grapes, we call this phenotype as angur. Now, when I joined the lab, this mutant was not characterized. That is, we did not know what was the gene that was responsible for this particular phenotype. So what I then did was I used a deficiency mapping approach. So in flies, we have different deletions available, and we know the chromosomal locations of these deletions. So what you do is in standard genetics, is you just take this particular allele and cross it with one of these lines, and you see whether they complement or not. So uh, the idea is if they do not complement, then that is probably the region where the particular gene is located. So in this case, uh, you can see that there are three different regions spanning this locus. And when you cross this particular mutant uh, with, you can see that the AP2 sigma gene is located here. That is common to like all these three deletions. So when you cross it with any of these three deletion lines, you will not get viable animals. So they will always be lethal and they will fail to complement, which means that your uh, particular gene or particular allele lies somewhere in this region. So firstly, we do a broad kind of a screening where you have these bigger deletions, and then you roughly locate the region. So from here, we kind of located this particular region, which had like different candidates. You can see that there are like almost 10 different candidates out here. So the next step would be to take mutants from all these 10 to 11 genes and do a candidate gene screening. So when I did that, I found that a particular mutant or mutation in this locus, the AP2 sigma locus, did not complement Angur. So that is where we came to know that this particular allele is actually an allele of sigma 2 adaptin. So to further confirm if this was really the case, we looked at the transcripts, like the AP2 sigma transcript in the different combinations. And we found that compared to the control in all these mutants, the levels of the AP2 sigma transcript was severely reduced by many folds. And to functionally prove that it was indeed the case, what we did was in this particular mutant, when we put back AP2 sigma transgene, we were able to rescue the phenotype. And also when we knock down the gene, you have a similar kind of effect. Now, how does this actually happen? I'm going to show you in the next slide. So in flies, we have a binary UAS GAL4 system. So this is actually borrowed from the yeast. So the yeast transcription factor is called GAL4, and that binds to something called as the upstream activated sequence. So there are two components to this. So you have a fly, or say, let's say this is a female fly. So you have the promoter sequence of a particular gene that is cloned upstream of the GAL4 encoding DNA. And in the other fly, you have the UAS sequence downstream to which is your particular gene of interest. Now, and this could be a transgene, this could be an RNAi construct. And so if you have an RNAi, you can use this to knock down your particular gene. So when you cross this, say, and if this promoter sequence is, say, a neuronal promoter, so you can achieve a tissue-specific knockdown. That is, you knock down this particular gene only in, say, neurons. And then, of course, you see what is the consequence of that. So in the previous slide, what I showed you was something as actin 5C. Now, actin is an ubiquitous protein. So if you use an actin promoter and knock down this particular protein, you're basically knocking down AP2 sigma in the entire animal ubiquitously. And then you see what the phenotype is. So these are some of the genetics that is available. And then there is something called as FLP, or flipase mediated recombination. Now we know that a recombination happens in meiosis, but you can induce uh, recombination to take place in mitosis, and that is with something called as the Frt flip method. And what you have here is a sequence called flipase recognition target, FRT. So if you have this sequence in the chromosome, and then if you come with an enzyme that is called flipase that recognizes these sequences, it will induce a recombination event between the two FRT sites. And as a result, what you have is you have a series of clones, wherein you have one clone that is a homozygous mutant, and you will have two heterozygotes, and you will have one which is a wild type. So this is particularly important when you have a mutation which is lethal, and I'm going to show you that in the next slide. So this particular mutant Angur was lethal. So you basically don't have a homozygous viable animal. So then the question is, if you don't have an animal in the first place, how do you actually study it? Because a dead man will not tell you the story. A dead man tells no tales, right? So, so the beauty of this genetic trick then helps you to study the function of this particular gene. So this is a wild type fly, and not a wild type. Actually, it's the FRT with the mutant Angur7. And here in this case, we made clones in the eyes. So what happens here is that you've got the animal, which is viable, but the eyes are homozygous mutant for this particular allele. So that's the beauty. You have a fly which is uh, heterozygous, but the eyes of this particular animal is homozygous mutant. And then you basically can now study the function. So this is a bright field image. Then we wanted to study in greater details what really happens. So we looked in using a scanning electron microscope. Of course, this work was done in collaboration with Dr. Praveen Verma at NIPGR New Delhi. And you can see that the wild type has these 700 to 800 structures called omatidia. And these omatidia are separated by these bristles, which are like placed between each of these omatidia. Now, when you look at the mutant, you first of all see that the size is very much reduced. And you can also see in the inset that the morphology is very much awkward. You don't have those fine rounded structures that you see in the wild type. Furthermore, you don't really have those bristles that separates the structures. So that was an indication that something is really kind of wrong. And then just to make sure that it is this phenotype is because of the sigma 2 adaptin, when you provide sigma 2 adaptin back to the eyes in these animals, you see that you are able to partially rescue the phenotype. So we were interested in further understanding, like at the ultrastructural level, what really happens. And so this is a transmission electron micrograph. So you take a cross section of these ommatidia, and what you see here are, so these are called as the rhabdomeres, and there are 8 such rhabdomeres, R1 to R8. Then you can see only 6 or 7 of them. So this is a wild type rhabdomere. If you look at the mutant again, you have absolutely nothing. So this is just like a hollow space here. So there are no rhabdomeres. Again, you put back the transgene, and you're able to rescue the phenotype. So that tells us two things. That is, one, that it is indeed an allele of sigma 2 adaptin, because when you put back the gene, you're able to rescue the phenotype. And secondly, what I'm not showing you here is also we did some functional assays uh, using electroretinograms. That is, we tried to figure out what is the response of these flies to light, and we did find that they could not really respond to the light. So that again tells that there is a functional problem. And the reason you look at eyes in the first place is that, so you've got synaptic connections also in the eyes. So you have the retina, and then you have the lamina below that, and then you have the other structures that, and there are neurons connecting them. So this is a coarse way of actually first figuring out if there is a defect. And if there is a defect, then you can study further in the neurons. So this was our first study, and we found that yes, indeed, there is actually a problem. And of course, this is what I already talked to you about. So now we wanted to look at the terminals or the synaptic boutons. So I'm going to, in most of the slides, you will see this image, and that is just basically to guide you what we are actually looking at. So this is the entire larvae, this is the ventral cord, and the green one are the muscles, and there's something in red that is actually the neuromuscular junction, and this is exactly what you are seeing here. So this is the control. You have those nice varicosities. It's like a bead of string, very nicely arranged. But if you look at the mutant, what you'd actually see is that you have those much smaller structures, and they are a lot more kind of clumped together. And this is actually resembles a bunch of grapes, and that's from where this name Angur is actually derived. So now you see that there is a definite morphological defect in these mutants. So we then quantified a few different parameters. That is, we counted the number of these rounded structures, and we did find that the mutants, all of them have significantly more number of these boutons. And of course, when you put back the transgene, you're able to rescue it. And we also quantified the area of each of these rounded structures, and we find that, of course, in the mutants, this is very much reduced, as is obvious from the image. And of course, when you put back the transgene, you're able to rescue the phenotype. So the message from this slide is that, of course, when you have a defect in endocytosis, when you have a defect in the AP2 adapter complex, you end up having a defect in the synaptic morphology. Now, we wanted to further look at what happens if you also knock down the other subunits. Now, this in the previous slide, what I've shown you is this is a mutant. So we wanted to see what happens you knock down the other subunits. Like, so I showed you in the previous slide that there are 4 different subunits: the alpha, the beta, the mu, and the sigma. So what happens if you knock down the other subunits, and we have a similar kind of a phenotype. So that is, we say that knockdown of any of the other AP2 subunits phenocopies the sigma 2 adaptin mutant phenotype. Furthermore, we also knock down an enzyme that is PI4K3 alpha that converts the PIP2 to PIP3 in the membrane, and we again have a similar kind of phenotype. Again, this is the quantification, the same kind of quantification, and you have a similar kind of result. This was performed at 2 different temperatures because the GAL4 is pretty much effective at higher temperatures. So we used the 2 different temperatures, and it is actually the case in both the temperatures. All right, so now what we wanted to then do was figure out if this is required specifically for the AP2 complex, or it is also required for other stuff. So again, this is the neuromuscular junction. We looked at the alpha adaptin, the beta adaptin levels, and we do find that they are drastically reduced. And then you are able to get back the levels when you put back the transgene. Um, we also then looked at the levels of clathrin. And as I said, AP2 is a major adapter for clathrin, and we do find that clathrin is also reduced at the terminals. Further, we thought we should do some protein analysis to look at the total number of proteins. So this is a Western blot. And what you do see is that in the 2 mutants, you have almost nothing of alpha adaptin and beta adaptin. But for the clathrin, you do have a reduction. So it's almost like 50% reduction in this case. But you have some amount of clathrin left, and that tells you that when you don't have AP2, there are other mechanisms, that there are other proteins that can actually take care of clathrin stability. So there are other modes of endocytosis that is kind of still operating. And of course, alpha actin here is a loading control. So, so far what I've shown you is that the sigma-2 adaptin, the smallest subunit of the complex, is actually important for keeping the complex together, and it is also required for the stability of clathrin. So there's another structure that you have. You have those microtubules and microfilaments within the synaptic within the synapse. So you have the cell body, and then you have these tracks. These tracks are made up of microtubules and microfilaments, and they extend right towards the synaptic bouton. And why are these structures important? Because there's a lot of trafficking that takes place from the cell body to the synapse, or even like retrograde transport from the terminals back to the cell body. So there's a lot of communication that is happening here, and these microtubule tracks actually function in helping this transport. So, and again, if you look at some of the classical mutants, in this case, I'm showing you one of the mutants, which is called DAP160. Again, this is also a protein that is involved in endocytosis. So compared to the controls, when you look at these mutants, you see that there are kind of breaks. So this is a continuous structure. You have those rounded loops. So this is a labeling for a protein called as Frt, MAB associated protein, and you find those wonderful loops in these control animals. But in the mutants, first of all, you have an interruption. So you can see that there are breaks in between. So these tracks, or the tubulin based cytoskeleton is not continuous. There is a break. And this is exactly why you also have defects in synaptic morphology. So what we wanted to then see was whether this was also the case with our mutants. And when we did a similar staining for the Angur mutant, we did find that there are, towards the distal end, you have got these breaks. So if you see in the control, you have these looped structures, but in the mutants, you do get these breaks, which are like indicated with an arrow. And then again, of course, you put back the transgene, you're able to rescue the particular phenotype. So as a mode of quantification, what we did was we counted the number of boutons that have these kind of loops. And we found that in these mutants, we have a severe reduction in the number of loops compared to the control. And again, we were able to, of course, rescue the phenotype. So so far I've shown you what are the different defects that the sigma-2 adaptin mutant has. So we then wanted to see at the functional level what really happens. So we then resorted to electrophysiology, and the way this experiment is done is that you have a recording electrode that is poked into the muscle, and then of course you record these traces. So these are the spontaneous vesicle fusion events. Every time a vesicle fuses, so I showed you in the previous diagrams that you have those synaptic boutons, you have the neurotransmitters that being released, and then of course there are receptors. So this is a neuromuscular junction. So the receptors are in the muscles. So when the receptors get those neurotransmitters, uh, they'll show some activity. So that activity is basically what is shown in these traces. And these are called as the spontaneous fusion events or the miniature junction potentials, and that happens all the time. And so this is the kind of response that you get. But if you then take another stimulating pipette. So in this case, if you remove this brain, so the brain is basically sending the action potentials for the uh, muscles to depolarize. So in this case, you remove the brain and you take those nerve fibers in a stimulating pipette, and now you control the stimulation that you can actually give. So you basically inject current into these nerves, and then you see what is the response of the muscles. So when that happens, you get something like a big spike. And this is called as the evoked junction potential, wherein you have a synchronized vesicle fusion. So here, this is a very random event, but here this is a synchronized fusion event when you actually inject current, and then you see what is the response. So we did similar kind of experiments with these mutants, and what we did find was that the spontaneous uh, miniature junction potential amplitude as well as the frequency was more in the mutants. And this was again similar to identical to the alpha adaptin RNAi knockdowns. So this was the minis, and then of course when you do the evoked synchronous fusion test, you have a similar kind of there is a reduction in the mutant and the alpha adaptin RNAi. Of course, we could rescue this by putting the transgene back. So again, we quantified. So we quantified these amplitudes, and what we did find was that there was a significant reduction in the amplitudes of the mutants or the knockdowns. And again, we could rescue back these parameters by putting the transgene. Then we looked at the quantal content. So quantal content is a ratio between the evoked potential divided by the mini amplitude. So when you do look at this ratio, you find that there is a significant reduction in the quantal content in all these mutants. And again, in the rescue, you can bring it back. Now, this was performed by an electrophysiologist colleague of mine, Zishan. And what the quantal content actually tells us is it tells you the number of vesicles that are being fused per action potential. So you have a rough idea of how many quanta of vesicles are actually fusing per action potential. And so we now know that there was a defect in the quantal content as well in these mutants. So we then went ahead and tried to figure out what really happens if you give a high frequency stimulus. So in one of the diagrams, I did show you that there are these synaptic vesicles. So if you give a high frequency 10 hertz stimulation, what happens is that you basically deplete all the synaptic vesicles. And then what happens is that uh, you would not really get any signal. Then you give a period of rest for some time. So when you give this period of rest, there is a refilling of the vesicles, and then you can kind of get back the same amplitude as you see in the starting with respect to the first action potential. So when you look at the mutant, or even for that matter, the RNAi, what happens is there is a gradual reduction in the amplitude over a period of time. And even if you give a period of rest, they are not able to recover back to the wild type levels. So you can see that these are the wild type levels, which they recover after the period of rest, but these in the mutant animals, you don't see any such recovery. And this is again similar in the case of when you knock down the alpha adaptin RNAi. And in the rescue, when you put back the transgene, you almost get back to the wild type levels. So this is what really happens. So we try to understand like what could be the actual reason behind that. And for that, we again collaborated with NCBS, and we did electron micrography of the synaptic boutons. And what you see here in cyan is the presynaptic part. You have those round vesicular structures. This dark one is a mitochondria here. And again, in the mutant, what you do see is that you have those bigger vesicular structures. You have something like a big thing bulging here. You have some bigger structures out here. So that kind of explains, if I go back to the previous slide again, you have those bigger amplitudes. So you have so now you can kind of appreciate what these bigger amplitudes mean. So these mean that you have got bigger vesicles, and they, of course, will have more neurotransmitters. So once they are released, you have these kind of bigger spikes. And also the fact that they would release more neurotransmitters, you also have these events happening more frequently. So that kind of explains what you see in the previous results. And so we do kind of understand now that this particular adapter complex, the AP2, could be actually involved in regenerating synaptic vesicles, probably from this kind of bigger structures. Now, when I say this kind of bigger structures, I'll come back to that in this slide. So the classical method of endocytosis, as I said, is you'll have these vesicles come and fuse, and they need to be regenerated back, and then, of course, they again reform the vesicles. Now, there are different models of by which these vesicles can actually fuse. So the one is called the kiss and run. So what happens in kiss and run is that you have these vesicles, they come and intermittently fuse with the presynaptic membrane. So they don't totally fuse, they just partially fuse, release the neurotransmitters, and they immediately are recycled back. And the classical way is wherein you have a vesicle, they come and totally fuse with the plasma membrane, and then of course they have to be recycled back. The third method is called as the ultra-fast endocytosis, wherein you have the fusion, and then the bulk of the membrane is kind of recycled back. They form an endosome, and from endosomes you again have these smaller vesicular structures which are being converted into the synaptic vesicles. And there's also a bulk endocytosis, wherein you have again bigger chunk of the plasma membrane that is being released, and then again you have to regenerate vesicles from these bigger structures. So what we kind of figured out from the previous set of data, that is the functional data and the electron microscopy data, was that probably the function of AP2 is not very critical when it comes to generating vesicles from the plasma membrane. But when it comes to generating vesicles from these kind of structures under high frequency stimulation, uh, it actually fails to do so, because there is a severe defect in AP2 function. So that also means that to make those vesicles from these endosome-like structures, you actually need clathrin and the AP2 complex, without which you cannot really make that. So uh, that's kind of what we figured out, and that is again consistent with the kind of results that were obtained or that is reported in some of the other studies, and they all talk about bulk endocytosis and the requirement of clathrin probably from this kind of structures, but not from the plasma membrane. So, so to sum up the first part of the story that I've told you is that, sigma-2 adaptin regulates synaptic morphology. So if you mutate sigma-2 adaptin, you have some defect in the morphological phenotypes, and they not only facilitate basal synaptic transmission, but they are actually required for regenerating synaptic vesicles during high frequency nerve firing. They are required, I mean, the complex is required for the stability, for its stability, and also for clathrin. And it regulates synaptic growth by controlling the microtubule cytoskeleton. Of course, this was published in 2016, and this is the reference. You could actually look back at it. So now coming to the second part of the same story. So as I've shown you before, like the mutant was called Angur because it had a aberrant synaptic morphology phenotype. And if you look back at some of the other mutants of the endocytic pathway, like endophyllin or synaptogenin, all of them have this similar kind of phenotype. So the question was, what actually goes wrong in these mutants, and what is the reason for this kind of aberrant synaptic phenotype? So again, that's what the question was. You have this beautiful control animal. There's all synaptic boutons are like arranged in a beautiful beads on a string. And here in this case, you can see that that is totally disturbed, and you have the smaller structures and this kind of a weird phenotype. So the question was, why is this happening? So to look at that, what we wanted to first look at was the growth signaling pathway. So this is not as simple as it looks here. I have kind of made it oversimplistic, but there's a lot of crosstalk that is happening, and there are more than just these two pathways. But for the sake of this presentation, I've just tried to show you two pathways. And one is the BMP signaling pathway, and the other is the MAP kinase pathway. So in the BMP signaling pathway, what happens is you've got MAD, which is actually phosphorylated, and then it forms a complex with COSMAD, which is called media, and this is then transported from the nerve terminals to the nucleus. So that's a retrograde transport, and then of course, once in the nucleus, it activates the transcription factors, and then you have various growth promoting genes that are being transcribed. On the other hand, you have a MAP triple kinase pathway, and that is, there is a protein called Valenda that activates a Jun N-terminal kinase that activates transcription factor FOS in the nucleus, and that leads to the uh, expression of your other growth promoting genes. And there is this E3 ubiquitin ligase called HiWARE, which is at the intersection of these two pathways, and apparently it controls both the signaling pathways, because Valenda is a substrate for the HiWARE. So what it does, it sequesters Valenda. So if it basically degrades Valenda, you have this branch of the pathway that is kind of being controlled or kind of being silenced. So if you don't have HiWARE, or if you look at a HiWARE mutant, you have more levels of Valenda. And if you have more levels of Valenda, this particular pathway then gets hyperactivated. On the other hand, HiWARE also is known to interact with this particular protein called media. So if HiWARE binds to media, the MAD is basically free. So what happens is MAD cannot form a complex with media, and if MAD doesn't form a complex with media, you cannot activate the downstream signaling pathway uh, that activates growth promoting genes. So HiWARE mutants have aberrant synaptic growth, and that is already documented. So we then wanted to look at first the BMP pathway, and the way to look at it is like we wanted to look at the levels of phosphorylated form of MAD. Again, you're looking at the neuromuscular junction here. In the control, you really don't see any of the staining for MAD, but if you look at the Angur mutant, you see that the levels of MAD are really high in these synaptic terminals. Again, when you put back the transgene, you're able to get back to the control levels. So for sure, the BMP signaling is upregulated in this particular mutant. Then we also wanted to see what happens to the other branch of the pathway. So in this case, you are looking at the ventral nerve cord. So this is this portion of the larva. And again, what you see here is in the mutant, you have those uh, staining pattern for Valenda, which is really, really high. And when you put back uh, AP to sigma transgene, you kind of get back to the control levels, or even if you put back a HiWARE, you get back to the control levels. So that shows that, Valenda is also high in these mutants, and that's why the MAP kinase signaling pathway is also getting activated. So both the pathways apparently seem to be activated in these mutants. And what we then next did was we looked at the other downstream factors, as I showed you in this slide. So these are the different proteins that are part of this pathway. And if you want to really understand the function in a pathway, you then try to basically uh, manipulate some of these players, and then you see what really happens. So we were talking of these branch of the pathway, and then the idea was if you can uh, say block either JNK or block either FOS, what really happens to the growth. And that is exactly what we did. So we expressed a dominant negative form of the JNK and the dominant negative form of FOS. So that is essentially you are disrupting the function of these two particular proteins. And what we do see here is that compared to the mutant terminals, which are very much uh, clustered here, you see that there is some kind of a partial rescue, though the rescue is not 100%, but you definitely see some kind of rescue. And again, this is the quantification. You see in the mutant that there's very high number of boutons, but in the two rescue cases, you see that there is a significant amount of improvement when you compared it to the rescue animals. And of course, this work is presently being carried on by Manish Dwivedi and Isar Bhopal. So, so to sum up the second part, what I've shown you is that the sigma-2 adaptin regulates synaptic morphology by modulating the BMP pathway. So there are more experiments that are happening right now to understand how exactly the BMP modules are regulated. So there are these BMP receptors which are in the presynaptic membrane, and then of course we're talking of endocytosis. So there is a good possibility that these receptors are not being endocytosed, and that could be one of the reasons why the BMP pathway is hyperactivated. And what I've shown you here is WEND or Welenda is actually upregulated in these mutants, and that leads to the activation of the transcription factor FOS, and in turn the MAP kinase pathway. So with that, I think I'll move on to the third part. And okay, before going that, I began with asking these three questions, right? So what was the role of AP2 in presynaptic vesicle recycling? And yes, we did find out that it facilitates basal synaptic transmission, but then has a minimal role in synaptic vesicle retrieval at the Drosophila neuromuscular junction. The second question was, what does it do? Or does it have an exclusive role in vesicle recycling, or is it required elsewhere? So the results indicate that it is absolutely required for regenerating synaptic vesicles under high frequency nerve firing, and also for stabilizing clathrin. And thirdly, how is the defect in endocytosis linked to aberrant NMJ growth? And we did find that defective endocytosis perturbs growth signaling pathways, resulting in NMJ, aberrant NMJ growth. And of course, the two pathways under investigation right now are the BMP and the MAP kinase pathways. So that kind of sums up uh, my PhD thesis work. And now I'll just switch gears and move on to another interesting study that also I did during my stay at ISER Bhopal. And this is more related to diseases. And so this is something to do with amyotrophic lateral sclerosis. And I'm sure this image looks very familiar. So this is the ice bucket challenge that kind of went viral, I think around 2014, 2015. And this was done to raise a lot of money, and that was uh, then channeled to do research in ALS. So what really is this ALS, and how does it actually affect the muscles? So I showed you most of the slides previously where we looked at the neuromuscular junction, and you have those neurons that have those long axons, and they send their uh, projections, and they innervate the muscles, right? So what happens in these diseased conditions? So in the normal condition, you have a healthy nerve, and then it innervates the muscles, and it's functional, as it's evident from uh, uh, the electrophysiology recordings. You see that absolutely normal. But what happens in amyotrophic lateral sclerosis is that, first of all, you have damaged nerve. You don't really have the continuity. In most of the cases, you will have degeneration happening towards the terminals. They apparently don't innervate the muscles that well. And so what happens is the muscle loses contact with these neurons. And so functionally, uh, they're not able to respond to the stimuli. So this manifests in the fact that uh, you have muscle wasting, and sometimes you're not able to say hold an object properly. You just don't have the stimulus to perform that motor activity. And so this is in brief what happens in ALS. Now, the next important thing is how does it actually happen, or why does it actually happen? So ALS mechanisms are very varied. You have got different mechanisms. A few of them are listed here. You've got sometimes you have disturbances in protein quality control. The protein gets degraded, or it fails to get targeted to the endosome, lysosome, or autophagosome. So you have got uh, uh, proteins which should actually be degraded, but they are not. So they tend to remain around at the synapses, and then that kind of hinders the whole process of synaptic communication. Sometimes what happens is you have the microglia which are hyperactivated, so they release a lot of these free radicals or reactive oxygen species, and they are again detrimental to the neurons, and they result in cell death. You have these other problems where there could be defects in the lactate transporters. You don't have enough energy that is being that is reaching the synapses, and so again they kind of die out in time. Sometimes what also happens is that you have neurotransmitters that are being released. Now, the neurotransmitters that are not being used up need to be recycled back, but sometimes due to defects in the recycling machinery, you have the neurotransmitters that remain in the synaptic cleft for a longer time, and that results in excitotoxicity, and that again kills the neurons. There could be defect in the cytoskeletal proteins. So I showed you previously that you have those microtubules and microfilaments that runs along these axons. So if there is some kind of a defect in those cytoskeletal proteins like dynein or dynactin, again, you don't have proper trafficking taking place, and then this kind of affects the motor neurons. And lastly, it can also be due to disturbance in RNA metabolism. So there could be problems with the RNA binding proteins, which are somehow getting sequestered, and they are not able to uh, bind to the target proteins, and then you either lead to less translation of the target proteins, or it could be that some proteins are highly translated at the synapses, and that lead to this kind of defect. So as you can see, like there are varied reasons for ALS to actually happen, and it's not just familial. Like 10% of the cases are familial, that is, it's hereditary, but 90 to 95% cases are sporadic. So even if you don't have a family history of ALS, you still have a chance of developing ALS. So that is a serious issue. Um, then of course there have been many genes which have been implicated for ALS, and so they are grouped into different subcategories. You can see that there are RNA processing genes. There is something called to do with glutamate excitotoxicity, oxidative stress, protein degradation, chromatin remodeling, endosomal trafficking, mitochondrial dysfunction. So there are many more. So you can see that there is a very wide spectrum that is associated with ALS. So what I'm going to take you through in the next couple of slides is our work with this particular protein called senataxin or SETX. So SETX is RNA binding protein, and this is the human protein, and you've got different domains like a protein interaction domain, ATP, JTAP binding motif, and then you of course have the most important DNA RNA helicase domain. So this is the domain with which it uh, uh, binds to the different RNAs, and then probably regulates its function. So when we look back at the literature, there are from different studies across patient samples across the world, um, there were a couple of mutants that have been identified, like the T3I, R2136H, like you have an arginine to histidine mutation at 2136H, and then you have a 1363 That is, there is a stop codon that is being introduced at the position 1363. So what happens is you get a truncated protein, or you also have something like a threonine to isoleucine at the third position. So there are different mutants, and these have all been like reported in human populations. So what we then did to study the function of this protein in fruit flies, uh, we made different transgenes and we express this particular protein in the Drosophila neurons. And we then wanted to see what is the consequence of expressing these uh, human protein in flies. And what we did find was if you express the wild type or the two other mutant versions in Drosophila neurons, you find that it leads to NMJ structural plasticity. What I mean by that is that you've got more number of synaptic boutons, and you can see that there is a parent bouton here which is surrounded by these small structures called as satellite boutons, and this is the case with both the mutants as well. So the wild type as well as the control, as well as the mutant forms of these particular overexpressing animals show increase in the bouton number and the bouton area. So this a similar kind of quantifications that I've shown you previously, and yeah, so this is something that we figured out, and then we wanted to next see like whether that is because of a perturbation of any of the signaling pathways. And here again, you're looking at the neuromuscular junction. And again, the two most studied pathways, as I said previously, are the BMP and the MAP kinase pathway. When we looked at uh, the the PMAD signals in these animals, we saw that compared to the controls, the signals were higher in these wild type or the mutant animals. And again, in the wild type and the mutant, you have the protein called high-wire, the E3 ubiquitin ligase, which has actually gone down. So this shows that when you express this particular protein in Drosophila, what happens is you perturb the signaling pathways, mainly the BMP and the MAP kinase pathway, and that results in aberrant synaptic growth. Um, what we also did was we expressed the truncated protein, and we expressed the truncated protein in muscles, and this result was really interesting. So what you see here is the muscle that is shown here, the round structures, the nuclei, the muscle nuclei, which is labeled with DAPI. And then you have this protein called ELAV. So this is an RNA binding protein, and so this is actually a protein that is present in neurons. So it is not supposed to be present in the muscles. As you see in the control animals, you don't have any signal for ELAV. But when you express this truncated form of protein in the muscles, you see that ELAV expression is very much strengthened in this case. And with this, what happens is we kind of proposed a model wherein uh, human synataxin somehow is relieved of the repression. So in the muscles, you have the ELAV mRNA that is somehow translationally repressed. But when you express this human synataxin, what it probably does is it kind of interacts with other proteins that are known to repress the translation of ELAV in the muscles. That interaction is disrupted, and then you have ELAV being kind of expressed. So this is an interesting finding because, as I said, the RNA binding proteins and one of the key causes of synataxin dysfunction. So, and we probably, though this is not in the human or the human cell lines or vertebrate system, yet I would say that in a heterologous system, this is an important finding because you kind of show for the first time that uh, you perturb the translational machinery, and that could be one of the reasons why the the patients having this kind of mutation, which removes the DNA RNA helicase domain, kind of fails to work properly. So to sum up what I've shown you in the synataxin story is that the expression of the human synataxin in Drosophila motor neurons induces some morphological plasticity. And then, of course, you have the human synataxin when expressed in motor neurons alters the BMP or the MAP kinase signaling pathways. And when you have the truncated form of synataxin expressed in muscles, it alters the translational profile of ELAV, that is an RNA binding protein, and actually it might be altering many more other RNA binding proteins. And again, these two stories are published, and this is the reference for that. So with this, I would like to just have one slide telling you what I'm doing in my postdoctoral work. So the previous work, what I've shown you today, is all from my doctoral work. And um, as was pointed out previously in the introduction, that in my postdoc, I'm looking at the postsynapse. So you have those receptors that are in the postsynaptic membrane, or say for the Drosophila and MG, it's the muscles. So they need to be, they are synthesized, they are translated, they have to go through the ER and Golgi network, and then they have to be, these receptors have to form functional tetramers, they have to be trafficked or to the surface of the muscle, then they have to be kind of clustered and sent to these synapses, and then that's where they are kind of functional. So the study basically involves what are the different mechanisms by which these receptors are stabilized, what are the different mechanisms by which they are trafficked to the synapses, and how does the clustering actually take place. And there are interesting studies where you found auxiliary subunit proteins that help in the surface delivery and the clustering of these molecules. Of course, that story is probably for another day, but that is something that I'm actively looking at presently, and trying to understand the postsynaptic differentiation and how you actually uh, sculpt synapse, and more from the postsynaptic side. So with this, I would like to acknowledge the people who've helped me during my PhD and all who are helping me now in my postdoc. So from IISER Bhopal, my mentor, Dr. Vimlesh Kumar, and this is the bunch of people who've been associated with the work. Um, Sishan, Abdul, Manish, Bhagwan, Asim. So they've been mostly involved with the sigma to adaptin work. Sri Krishna, Namrata, and C have been involved with the synataxin work. Abhinandan was recently involved with a part of the sigma to adaptin study. Akil is the person who takes care of the flies and make the food. So he's an important resource. And most importantly, Dr. Vimlesh Kumar is a wonderful mentor, and well, I've spent like 6.5, 7 years in Bhopal, so it was a learning experience working with him and working with fellow colleagues. And at the National Institutes of Health, I have a wonderful mentor in Dr. Ella Serpe. She's very encouraging and um, very supportive. And you've got a bunch of colleagues. They're all postdocs: Rosario, Tahi, Thol, Lindsay, Wenjay, and Tom Brody. Tom Brody is here. So he's a veteran, and he is also the one who maintains a website called the Interactive Fly. Um, and well, people familiar with Drosophila would definitely know him. Uh, Peter, of course, takes care of the lab, and uh, the current funding is from the NIH intramural funds. And for my PhD work, I was funded by the Ministry of Human Resource and Development, the Department of Biotechnology, the Department of Science and Technology, and the ICERB intramural funds for the experiments. So thank you so much for your patience and for listening. And if you have any questions, I'll be very happy to answer.
Thank you, Soumitra. Now the session is open for questions. Participants, if you have any questions, you can post it in the chat box. Mhm. Okay, I think now at present there are no questions. Okay, maybe later. Okay, yeah, there is a question coming. So he's actually talking about the particular slide wise. In slide
Okay.
21, can you see that?
I can see the question. It says slide 21 alpha adaptin
Yeah, yeah,
RNAi
yeah.
current is not going back to the basal level as like the other variants. Why is this happening? So this is an RNA lane, and it could actually have possible problems. The knockdown may not be as efficient. So yeah, that could be an essential. That could be a reason, I would say. The other ones are the mutants. So you have it like the mutant is like it's a mutant in the whole animal is a mutant, but if you look at the alpha adaptin RNA, you're just knocking it down in the neurons. So there could be other things happening from the muscle side or from the glia, and also the knockdown could not be 100% efficient. So there could be that kind of a problem. Yeah.
Any more questions? Yeah. Yeah. And one more question came, Soumitra. Has there been progress in studying the genetic aspects of diseases?
Yeah, I mean, using the fruit fly, yes, there have been many studies using human diseases because, as I said, like 70% of the diseases that that occurs in humans, you have a particular homologue in flies. And the advantage of using flies is that in humans, you might have different redundant genes. So if you knock down one, or if there is a mutation in one, the other proteins can actually take over. But uh, in flies, most of the cases you just have one particular gene, so you usually get a phenotype when you mutate that.
Yeah.
So yes, that happens. And then he's asking, rather than just the superficial causes like defective proteins.
Difference.
Okay, so this was just one example that I showed you today with senataxin. So this is known to uh, have a problem with RNA binding, and then you have this kind of things. But you have other diseases in which people are actually figuring out the mechanistic reasons behind what is actually happening. So there's a lot of disease work that is actually going on. So and since uh, as I said, like there are functional homologs, it's very easy to actually figure out the mechanisms. Yeah, but one has to be very careful because the disease work is uh, it's demanding, or it's challenging, I should say, and it's easy to come out to have wrong conclusions. So that is one major problem, I would say, in the field. So one has to be very critical with, when you are studying diseases. Yeah. Does that kind of answer your question? I hope it does. Okay.
Okay.
I think the screen froze there, probably. Looks like.
Yes.
Uh,
Yeah, nice presentation, Soumitra. You have a lot of data.
Thank you. So these are already the previous work from my PhD. So right now I'm like kind of working on the postsynaptic side. So yeah, there are certain nice findings, but since I don't have a complete story, I thought I'll just talk about the work that is kind of complete.
Yeah.
Yeah.
What are these actually so-called gate signaling? So do you know any idea about gate signaling? Because in the first slide, I think you were showing like one side now passes the signal and another one receives it.
Yeah.
Here comes like a gate signaling, right? If I am
Yeah.
right.
Yeah.
But I know you didn't go through this today, but I just have some background idea. That's why I'm asking. So what are these gate signals? How they work in this case? Any
Okay.
idea?
I see.
No, have you worked on that?
No, I have not worked on that. I have not worked on that. But then there's a retrograde signaling that happens from the other side as well. So that is again a very interesting thing to look at. So currently the lab is actually looking at a retrograde pathway that goes from the muscle to the neuron, the other side. So yeah.
Nice. When did you join NIH?
I joined in 2017. It was like September 2017. So it's about to be 3 years now.
Yeah, almost. Good, good, good.
So you've been in the US for, I guess, quite a long time now.
Yeah, now in the USA for almost 6 year crossed actually. This, I mean,
Oh, okay.
what's the date today? Third, right? Third, fourth.
3rd July, yeah.
Maybe
Third.
3-4 days. I want to finish my 6th year.
Oh, yeah, it's a long time. That's nice.
Good year. What is your department there in NIH?
Child Health and Human Development.
Okay.
NICHD, yeah.
I think their screen is, I mean, kind of broken or.
I don't know. It looks like.
Hello, Chandan. Okay.
Yeah.
Can you listen? Ah,
Okay. Okay. Okay.
What's the time now? I think you are 1 hour ahead of us, I guess.
Yeah, yeah. It must be. Oh, it's like 2:30.
Yeah, for us it is 1:30.
1:30. Yeah.
Hello.
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 Okay. Sorry for the interruption. Suddenly there was some problem, some technical issue.
That's okay.
Okay. So it's 2:30 there, Soumitra.
Yeah, yeah. It's fine because now my circadian cycle has totally changed. Like,
Okay, good.
so it doesn't really matter.
Okay. So thank you so much, Soumitra, for sharing your informative talk with us. Okay, started with the basic, with the diagrammatic representation and all. It was so nice. Okay, the way you
Yeah,
presented,
thank you.
taken us into your doctoral discussion, and then later your postdoc thing also you have little bit touched. So it was very
Yeah,
nice, very informative session. Yeah,
thank you.
yeah. So we will conclude now with the vote of thanks.
Okay.
Okay. Respected Principal, Dr. E. Jerome Xavier, Indian Academy Degree College, Autonomous. Dr. Prakash, Scientist in University of Iowa. Dr. Soumitra, Scientist in National Institute of Health. Members of faculty, and all the student participants. A very good afternoon. Almost it is 12 now. I deem it a great honor to propose the vote of thanks. First of all, I would like to thank our chairman, sir, Dr. T. Somashekar, Indian Academy Degree College, Indian Academy Group of Institutions, and our secretary, ma'am, Mrs. T. Bharati, Indian Academy Education Trust, for their constant support. I would also like to thank our COO, Dr. Srinidhi K. Parthasarthi, for his support. I would like to extend my heartfelt thanks to our beloved principal, Dr. E. Jerome Xavier, Indian Academy Degree College Autonomous, for his continuous support and encouragement. I would also like to extend my thanks to our resource persons, Dr. Prakash and Dr. Soumitra, for making this event a memorable and a successful one. And let me thank all the participants for their presence and active participation. I would also like to thank our HOD ma'am, Dr. Prashanti, for her help and support. And all this can't happen without a teamwork. So we had a very nice team, including Professor Dilshad Begum, Professor Malati, and a special thanks to Mr. Chandan, the head of the system admin team, for his sincere efforts in helping us in arranging the webinar. I also thank all the technicians for their help. Once again, thank you, all of you. And already regarding the feedback link,